Welcome to the first episode of the Theoretical Neuroscience Podcast. I am the host, Gaut Einvoll. The guest of today's episode is Grace Lindsay, author of the popular science book, Models of the Mind, excellently describing the theoretical neuroscience field. Before I start, I thought I should talk a bit about why I decided to make this Theoretical Neuroscience Podcast. Well, since 2019, I have been making podcasts on science and other topics that interest me. The podcast is called Vett og Vitenskap. It's mostly in Norwegian, but with a few episodes in English. And I found out that I really enjoy making podcasts. So why a new podcast series focusing on theoretical neuroscience? Well, theoretical neuroscience can be said to be a sparse field, meaning that we are rather few people compared to the number of topics that we address. And only a few percent of neuroscientists would identify themselves as theoretical or computational neuroscientists. So students or researchers doing theoretical neuroscience often don't have a large community of other like-minded neuroscientists around them. Being a computational neuroscientist myself, I decided to make this podcast with the hope that it can help in contributing to a lively and generous theoretical neuroscience community. I have been inspired by the podcast Brain Inspired, focusing on research at the interface between neuroscience and AI, which I follow regularly. For me, it is much easier and enjoyable to listen to a podcast lasting an hour or two to learn about the new topic rather than reading a paper. I suspect it is the same for many other scientists. I will have some costs related to the production of the podcast that I hope to get covered. I prefer not to have commercials in the podcast, and I also don't want money issues to get in the way for people who would like to listen to the podcast. So for now, I have decided to simply make the audio version free, and this will be available on all major podcast platforms and also through the webpage theoreticalneuroscience.no. On this webpage, you will also find info about the episodes with links to relevant material. However, I have made a Patreon page where you can just subscribe and get not only the audio podcast, but also video version of the podcast as well as transcripts of each episode. But you can also watch these videos on the YouTube channel Theoretical Neuroscience Podcast. If you want to support the podcast, you can go to patreon.com/theoretical neuroscience podcast and choose a membership of your liking. For example, at the cost of $1 per month. Even if you're happy with listening to only the audio versions of the podcast, I hope that many of you will sign up as Patreon supporters to support the production of the podcast. I can assure you that I will not pocket any of the money for myself. Everything will go into the production of the podcast, mainly to help with the technical aspects. But now to the first episode itself, where I'm very happy to have Grace Lindsay as a first guest. Grace is a computational neuroscientist working at New York University, and she published a book, Models of the Mind, a few years ago, which gives a popular account of the history of theoretical or computational neuroscience, and also the problems that the field is interested in. I'm quite impressed by this book and like it a lot, and I recommend it for all neuroscientists, computational or otherwise. In the episode, we talk about the history of biophysical modeling of neurons, from the discovery of bioelectricity by Galvani 200 years ago, to Hodgkin and Huxley's modeling of the action potentials, and onwards from there. We further discussed the modeling of information flow and computation, starting with the seminal paper of Pitts and McCulloch 80 years ago, that also had a major impact on the start of the AI field. Next, we discussed some highlights from computational modeling of networks, for example, Hopfield's memory model, and the balanced excitation inhibition ID explaining the noisy firing of neurons in cortex. We also talk about the recent development where deep networks are used to investigate biological vision. This is Grace's own research field. And finally, we speculate a bit about the future of our field. Okay, here is Grace. Welcome to the podcast, Grace Lindsay.
Thank you. Happy to be here.
Yeah, so when I decided to make this new podcast on theoretical neuroscience, my first thought was that I should have you as a guest for the inaugural episode, because I think you've written this really excellent and impressive book, models of what is it? Models of the Mind.
That's right.
Yeah, and which then I think sort of both gives a very nice historical introduction to the field of theoretical neuroscience, computational neuroscience, and also it very much covers many of the problems that the field is now working on. So first, I mean, can I just why did you do this? I mean, you must have been an active popularizer maybe before because of quite ambitious thing. I think you were a postdoc at the time, so.
Yeah, I was just starting a postdoc, and yeah, during grad school I had done a decent amount of science writing on my own blog and other outlets, and then
Mhm.
I also had a podcast for a few years.
Yeah, that
That was about
unsupervised thinking
Yeah,
was it?
that's right.
Yeah.
Yeah.
Mhm.
That was about computational neuroscience and artificial intelligence and neuroscience and that sort of
Mhm.
thing. So I always kind of was interested in getting a broad view of the field,
Mhm.
and kind of thinking at maybe the more like meta science level of how it works,
Mhm.
and why mathematical models are a good idea. And so yeah, and then through the writing that I did, I got to know people who had written popular science books, and it
Mhm.
kind of became a more tangible, possible thing.
Mhm.
And then yeah, there was a publisher. The publisher I ended up using, Bloomsbury Sigma, was looking for more authors, and I had a conversation with the editor, and it kind of just snowballed from there to being an
Mhm.
actual book.
Wow. Yeah. So this is, I think, as well. Well, when I read this book, I thought this must. Well, it seems like it should be written by a person who's been like many decades in the field, and you were just a starter. So that was quite impressive. You also came up. There's a lot of metaphors that you have in there. So that about sort of like how to describe what's going on in neural systems. So you made. I haven't. Most of them I haven't heard before. Did you make them up yourself, or?
Yeah, yeah, for the most part. I mean, I knew, so, there are a lot of people who are in the field or in an adjacent field that have read the book, and I
Mhm.
figured that would happen. But also it's supposed to be a book that's accessible to a general audience.
Mhm.
So I wanted to make sure that, even if you didn't get the exact scientific description, that you
Mhm.
could capture the general vibe by
Mhm.
having
Yeah.
these metaphors. So
Yeah.
yeah, it's usually just kind of thinking about how I'm visualizing the concept in my head and trying to come up with
Mhm.
something in the real world and like the
Yeah.
normal everyday world that relates to that to kind of get the idea across.
Mhm. So yeah. So anyway, so I think just to start, I think this should be a book that, well, this is a book that belongs in the bookshelf of any computational neuroscientist and lots of other people who are also just interested in what's going on in the field. I think it gave a very nice and accurate description of what's going on and sort of like the main ideas and so on. So kudos to you. And I thought I should start with like going back I guess to the start of in some sense of I guess maybe computational neuroscience or even at the beginning of neuroscience with the discovery of electricity in the brain with Galvani. So what did
Uh,
he do?
Yeah. So he was an Italian scientist who was studying a lot of different things as scientists of that time did. They didn't really specialize yet. So he was studying biology and electricity and physics and all of those different things. And through kind of an accident in his lab, he discovered that if you apply electricity to a frog's leg, it twitches.
Hmm.
And then that, suggests that biological beings are using electricity in some way. They're using it to conduct signals or to cause movements. And so he kind of followed that up with more experiments of saying, yes, if we apply
Hmm.
electricity to different organisms, you can get responses, and that kind of set off. A long debate amongst people as to whether electricity is really used by animals
Hmm.
natively, or if
Hmm.
this was just something that kind of you could get to happen if you applied electricity, but it doesn't actually speak to how the body really
Hmm.
works. That was kind of the debate that he set up.
But one very concrete thing that got out of it was this novel Frankenstein, right?
Yeah, yeah.
Yeah.
So, yeah, because this was a time when, the boundary between different types of science was weak, and the boundary between a scientist and a layperson tinkering in their, in
Hmm.
their own home was kind of weak as well. The notion of electricity creating life or
Hmm.
reanimating things was spread throughout popular science, or through popular culture rather, and people were playing with some of these devices, like a Leyden
mm,
jar is a device
mm,
that can hold charge, but it's pretty simple and easy to build. And so people could play with these things in their home, and it really just spread through the popular imagination what
mm.
electricity could do and how it could relate
Uh,
to life and how,
yeah.
life is generated.
I even heard that people were traveling around with corpses, giving them electric shocks so that they could scare people in the audience. So it
I would
was,
be scared by that.
yeah, me too. But then, I mean, we maybe also heard about Volta. At least that's another Italian who has the unit for electric potential named after him, volts. But he sort of, even though he made batteries and stuff, he still didn't believe that maybe that the electricity was something that the muscles or the biological tissues or brain could make
Yeah, and it's
by
because
itself.
the nature
Hmm?
of the experiments were not ones where you could observe electricity in the body. It was
Hmm.
just this foreign
Hmm.
application of it. And so, without being able to observe electricity directly, without it being applied externally, there was just, for Volta, not enough reason to believe that
Hmm.
this was happening innately in the animal, that it
Hmm.
was just a response or a result of external electricity,
Hmm.
but didn't necessarily speak to what was happening
Hmm.
biologically.
Because you even mentioned in a book that this very important physiology book by some Müller or something, that he sort of that 1840 didn't really believe that electricity was that important to understand the body.
Yeah, so he was a vitalist. So
Hmm.
he said that it's not electricity that's part of how nerves signal and communicate, or that leads to control or animation. Um, but also, this vitalist camp is kind of the mindset that you can't describe what it is. You're not going to be able
Hmm.
to give a physical explanation to what animates the body.
Hmm.
And so, from if you're starting from that point, then yeah, you're not going to believe it's electricity, you're not going to believe it's anything. um,
Hmm.
and Mueller, particularly, was also on the side of like experiments where you apply the electricity, those are no good. What you want is just observation. You just want to be able to observe and see what happens and draw conclusions from that. So
Hmm.
yeah, for reasons similar to Volta, he didn't believe in the electricity, but also for these more kind of fundamental philosophical reasons, he probably wasn't going to
Hmm.
believe in anything, because his whole claim was that there's this life force, and that's the
Hmm.
answer, but the life force isn't definable in physical chemical terms or anything like that.
Hmm.
um, which is
Hmm.
kind of a difficult position to stomach, I think, if you're having a
Hmm.
scientific mindset towards something. You kind of believe that
Hmm.
you can figure it out, and that it's not just going to be, you don't have to just appeal to something that's by definition immeasurable.
Hmm.
Yeah, that was the state of kind of the study of life at the time, which is kind of analogous to the study of consciousness now, in a way, where
Hmm.
people kind of appeal to the fact that there's something that can't be measured. But yeah, he wasn't, Mueller wasn't the only person who believed this, but he was a prominent person who believed it.
Hmm.
But eventually the actual experiments with
Yeah.
electricity won out.
Yeah, but because I guess also this was like the early days of electric studies, so I guess it also a question about like amplifiers were so like difficult to have like the get the right signal to noise ratio. But soon, rather soon after, there was like this guy, Du Bois Raymond,
Hmm.
and also then Bernstein, Julius Bernstein, that was able to measure well clear electric signals from I guess muscles and brain cells and so on.
Yeah, so Bernstein was a student of Raymond, and yeah, they were developing the necessary technology to actually be able to pick up the very weak electrical signal that can come from nerves. They were still working in the realm of nerves, not even individual neurons, but bundles of
Mm,
neurons. But yeah, they were able to stimulate the neuron at one end, and then put the recording device at a different end, and be able to see
mm,
at some time point later that there would be this kind of electrical disturbance in the cell itself, and therefore that the cell was using electricity to communicate. It wasn't just the result of externally applied electricity.
mm. I said that Bernstein was he did his work I think in 1868 where he measured the action potential. So that was sort of like I guess it's 150 years ago or something
Yeah,
where they were
well,
able to do
that's
that.
when you think about it like that, it's like neuroscience is pretty young,
Yeah,
because
it
the
is
action
true.
potential is
Yeah,
kind of core to the whole
yeah,
endeavor,
it's true.
and
So what could you do before it?
yeah.
Yeah, so that's true. So And I guess also I think the first I just because our group we have been working on these extracellular signals and stuff, and I think the first extracellular signal that was measured from the brain was the ECOG, which was like 1870 or something, where they
Sure.
saw some
Yeah.
So that was also when they had the guys amplifiers, which were good enough so that you can sort of
Methodology is very important.
yeah. So and then for those, I mean, many of us in the field have learned about the integrated fire model, and then it's often referred to. You refer to this person Lapic
Hmm.
in I think 1907 or something. So what did he do?
Yeah, so he took, at this point, there was a lot of evidence of how neurons respond, or nerves at least respond to applied voltage, and the idea that they're using electricity was accepted. But his goal was to quantify it more and kind of come up with a description of what was happening that had some mechanistic truth to it. And so to do that, he focused on the question of, if you stimulate the neuron, it was known that you stimulate the neuron with some electricity, and the muscle or the neuron will have some response later on.
Mm,
And so his question was, if you apply voltage of different strengths, at what time point will the nerve or the muscle respond to that application? Because it was clear that weaker voltage led to a longer delay, and higher voltage led to a quicker response from the neuron. And so he wanted to be able to come up with an equation that captured that relationship. And to do so, he just kind of took very directly the mathematical framework of how people describe electrical circuits. So just
mm,
thinking of the neuron as its own electrical circuit, and using the same mathematical
yeah,
tools that you would use to describe, wires
yeah,
and things. He used that to describe the neuron in what's called an equivalent circuit model of a
yeah.
neuron. And so that just
That was a
means,
quite important conceptual advance.
yes, yeah. To say, because then it makes this very clear connection between the two fields, and all the tools that are developed in one can then be used in the other.
Mm,
And so yeah, he described the neuron as having a capacitor that could hold charge, which is played by the role of the cell membrane. And
mm,
then also he acknowledged, though, that it wasn't a perfect capacitor. It didn't hold all of the charge that was put on it. It had some kind of leak to it, and that was played by the role of a resistor in the equivalent circuit
mm,
model.
mm,
And through that, he was able to um, make this connection between how much voltage you applied and when the nerve would respond, by assuming that the voltage had to reach some sort of threshold in order to
mm,
respond. And so
mm,
he was able to kind of fit the data that he had with this equivalent circuit model.
mm. So then the response was then what we now call the spike or the action potential, but they didn't really know what exactly happened at this action potential. That's some ideas that were just like the membrane was short circuited or something and was just opened up and became a hole in it, or like a
Yeah,
electrical hole.
so Lepic's
Mm,
model definitely doesn't try to explain what the action potential actually is. He's just
mm,
saying there's a response that happens, and it happens at some threshold, and that's all that he was trying to capture. And yeah, so the actual kind of if you really like zoom in on the action potential, it has a very characteristic shape to it. The
mm,
dynamics of the voltage of the cell has a
mm,
very characteristic shape. And yeah, the theories initially, from people like Bernstein, were that the cell membrane just kind of dissolves a little bit, and that's how the difference in charge between the two sides of the cell membrane, how that weakens during the action
mm,
potential. But that
mm,
wasn't a very good model, because it doesn't describe some of the more kind of extreme features of the action potential shape. If it was
mm,
just that the membrane was getting a little bit looser and letting more things
mm,
through, you wouldn't get the shape of the action potential that's
mm.
actually seen.
One element of that theory is that there is a potential difference between the inside and the outside to begin with, so like in rest. And I guess this was sort of like this idea with the Nernst potentials that there's like if you pump up or if you have different or unnatural concentrations of some sort of these ions of sodium and potassium, I guess, and chloride in particular, then you get this membrane potential to begin with. That's so. I guess that was also another pioneer was this Nernst.
Yeah.
Yeah, that also put this. That also made this key contribution. And then, but then I guess next big milestone if you jump until like the 1950 or something, was the Hodgkin and Huxley, because they then took it a step further and aimed to, well, not only aimed to, they tried to, well, quite successful in explaining what actually happened when you get this particular spike. Because it's not just that the membrane breaks down, it's like more particular picture, right? About with ion currents, ion selective channels.
Yeah. So Hodgkin and Huxley had to do a bunch of experiments with these different ion types by taking neurons and putting them in different solutions that contained or didn't contain different ions to see which part of the action potential would change if there was not enough sodium or not enough potassium or something like that.
Mm,
And so through that, they were able to see that there is a lot of very specific ion concentrations at play, that it's not just kind of a generic charge calculation. It matters the type of ions. And so they therefore built a kind of new equivalent circuit model, still based on the same basic principles, but here they added these extra resistances to the model. And those resistances were meant to kind of model the ion channels that were specific to different ion types. And
mm,
also, those ion channels, it turns out, are voltage sensitive. They're voltage dependent. They'll change how resistant they are based on the voltage of the cell. So that
mm,
creates this rather complex system with a lot of interdependencies and a lot of interesting dynamics. And through that, you get the shape of the action potential that we know
mm,
of, where the voltage changes in both directions and kind of goes reverses its potential and comes back down and everything.
yeah,
And so their model, through having the right kind of ion channel dynamics, the right understanding of those ion channel dynamics, they were able to capture the actual shape of the action potential.
mm. And that at that point you needed computers, or at least like some hand cranked, yeah, to do the numerics, because it was just, you couldn't really do compute the consequences with pen and paper easily.
Even just for one single point neuron model,
Mm,
it already
mm,
gets too complicated.
mm,
And there's a quote in the book from Hodgkin doing those calculations, where he says that he's been surprised by the outcome of it. Like, he's putting in the
mm,
calculations and he's calculating the voltage at the next time step, and he thinks it's going to lead to like a full action potential, but it dies out instead. And just this idea that
mm.
our intuitions for these really complex systems are just, they're not very helpful. Like, we don't have
No,
intuitions for
no,
these complex systems.
exactly.
And that's
Yeah,
kind of the whole point of doing mathematical
uh,
modeling, is,
that's true.
you figure out the basic principles and you can write down the equations, but you
Uh,
can't guess how they're actually going to play out.
yeah, exactly. And that's, I guess that's one of the reasons why when you're able to do mathematical modeling, it's so attractive, because the rules are so small. I mean, you can follow the logic. I mean, you can trust the outcome if you do the maths right, even though if you don't have intuition about it, right? Because
Yeah.
it's such a, so that's much more difficult with like I mean, models in the words. That's, they're difficult to sort of to come up with really unintuitive things.
Yeah, usually you're
Hmm.
just kind of restating your intuition with words, whereas the
Something
models
like
can
that.
actually return
Yeah.
something pretty surprising.
Hmm. So then we were, in terms of modeling neurons. Then I mean, we still didn't have like a good way to, or we didn't sort of, so far the focus hadn't been much on dendrites, right? But then then the computational neuroscience hero Wilfred Rahl, I guess, entered the stage. So what did he do?
Yeah, so he just kind of continued on with this idea that neurons are electrical circuits, and therefore you can use the tools that we use to mathematically model electrical circuits. You can use them to describe neurons and build models of neurons. Um,
Mm,
and in the case of dendrites, he's associated with the cable theory, where you basically just treat dendrites kind of as wires that have some resistance, and but they can carry the current to the soma. And so by adding in the morphology of the dendrites, just according to the basic equations by which you would model wires in an electrical circuit, then you can capture some of the functionality of dendrites in neurons. And so, something that comes out of that is just this idea that the pattern of stimulation on the dendrite can matter. So, what order you're
mm,
stimulating different inputs, because they're physically located in different places, they can lead to different results at the soma, and
mm,
that itself can be involved in computation.
mm. And I guess this, with that sort of like, and so he also, and I guess also, if you, so well, if you look at the Hodgkin Huxley, what they contributed was one thing they had, like came up with this sodium and potassium channels that actually was sort of in the right form to explain this action potential generation and propagation in this and squid giant axons. But they also, the formalism they introduced was also then could be easily adapted to other ion channels. And I think now with this much, well, this cable equation description overall and this Hodgkin Huxley description of ion channel currents, we sort of have it's like that's sort of the backbone of what's called multi-compartmental modeling, or like biophysical detailed modeling in neuroscience. So the principles of that is sort of like, I guess, was established in the 60s or 70s or something.
Yeah, or even with Lepic as early as, 1907. But yeah,
Mm,
building on that by being able
mm,
to collect data from real neurons and then use
mm,
that to get the parameters for the model. But having
mm,
the kind of mathematical tool set to be able to put in almost anything you could find in a real neuron, you can pretty
mm.
straightforwardly put it into one of these models, like
Yeah.
extended models.
And then this was then you also needed some models for the mathematical expression for the synaptic currents. But after that, you could start putting it together in these large networks. And maybe the most well known of these large scale networks is this Blue Brain model that came out in 2015 from this group in Switzerland. So what did that model do?
Yeah, so that model, yeah, kind of took these basic ideas and just really went crazy on scale with them, basically. So collecting data about all different types of neurons in the rat cortex and the shape of the neurons and their properties about their membrane potentials and ion channels and all of that, along with how they connect to other cell types and all of that to create just a really large model of a bunch of interconnected, thousands of interconnected neurons, which are governed by millions of equations, basically, and just running that simulation, on supercomputers to be able to take these basic principles of how you model individual neurons and then say, can we from that build a model that's actually more representative of an area of cortex, a very small area of cortex, but still some region of cortex, kind of going from these small biophysical models to still biophysical models, but at this really large scale, which is for the most part a very technically challenging thing to do, both in terms of collecting all the data you need to get the parameters and then actually running the simulations on computers.
One, I mean, of course, these large. We are also involved in doing this. In this, we're collaborating with people at the Allen Institute for the mouse primary visual cortex model, and of course, it's very difficult to analyze the results. It's sort of like and well to deal with these these models. But there's one example maybe where which for a simpler system with this stomatogastric, what's called stomatogastric ganglion in the crab, with Eve Marder and collaborators. So they have sort of been able to study. I guess that's one of the best successes of sort of studying and modeling a particular network system in a living system.
Yeah, that's an example that's rare, in the sense that there is kind of a lot known about it. It's not a very large circuit of cells, and it's used to control digestion. But it's well characterized. Like the data has been collected on the cell types and how they connect to each other and all of that. And its function is pretty clear. It's supposed to be generating these oscillations. And so I would say it's not, that setting is not super representative of the challenges that face most neuroscientists, because usually you don't have the structure and the low level details that well characterized, and sometimes you're not
So how
even
many
quite
neurons
sure.
are there? Like 10 or 20 or
Yeah,
30 or
it's
something,
on the
or that
order of
on?
that.
Yeah.
Yeah.
Mhm.
So
Yeah.
yeah, and a lot of times you're not even that sure about what the function is
No,
of the
no.
neurons that you're studying. But yeah, there have been, because it's so well characterized, you can kind of say
Mm,
things with more certainty and
yeah.
derive kind of principles that could be
Mm,
relevant for
mm,
everyone, for whatever you're studying.
mm,
And so one of the kind of arguments that Eve Marder makes, is that the relationship between the structure of the circuit and its function is not that tight. Especially if you're really defining structure as kind of the pattern of connections between the neurons. She's shown that you can have the same connections. You can have the same circuit really, but based on the different neuromodulators that are present, it can display very different rhythms. And so the function of it changes based on the presence of these neuromodulators that don't really change the physical connectivity necessarily, but change some maybe properties of how the cell responds, or maybe like scale the weights of the synaptic connections to some extent. So yeah, so the idea that a single structure can have many different functions is one side of it, and then also the flip side is that the same function can be produced by many different structures. So different individuals in this species will have slightly different structural connectivity, but they all still are able to produce the rhythms that are necessary for the
Yeah.
digestive system.
Oh, a flip side, maybe for the not for the animal probably because it makes them more robust. That there are many ways to have the solution, but in terms of for the modeler, if you want to look for some kind of unique solution, it sort of is. It makes it more, and sometimes ill posed to makes it more difficult. But I guess it's also, I mean, if you sort of like this thing of doing this biophysically detailed modeling, if you go take a computation neuroscience course around the world, you typically are introduced to cable description and Hodgkin-Huxley, and you maybe do some simulation or build a model, and there are like these tools like Neuron, Genesis, and Arbor where you can do these things, and then so that's sort of like a standard approach, and then you typically decide what ion channels you want to have in it, and then you sort of have to choose these densities of these different ion channels, how they are spread. But I guess like some people have also started to look at maybe this is like you could go one level deeper, and maybe instead of modeling the densities of putting in choosing the densities of ion channels as parameters, you could sort of model sort of the transcription or whatever, sort of like how these are, because these are dynamically regulated to begin with, right? Yeah. So, because this subcellular level has not been so much, I mean, what's going on inside cell, that hasn't really typically been the realm of computational neuroscience. Not too many computational neuroscientists are
Yeah.
doing it.
Yeah, I think there are people who are applying computational approaches to that, but they don't fall under the category of
Mm.
neuroscience. And they're
Yeah.
maybe doing it in a more generic way for
Yeah.
how cells, regulate these processes in
Mm.
general, but without, the motivation in terms of how these subcellular processes
Mm.
lead to things that are particularly important for the
Mm.
brain and how the brain functions. Yeah,
Mm.
a lot of computational neuroscientists have ignored them, and that's the art of computational modeling, is that you ignore the
Mm.
things that you
Yeah.
think don't matter.
I think that. Yeah.
And so ignoring subcellular stuff is a pretty easy argument
Mm.
to make a lot of times.
Yeah.
But I think there can be areas where people turn to that if they think it's important for what they're trying to
Mm.
understand. And one area that I've seen developing, kind of spurred by progress in artificial neural networks and thinking about how those learn and how that compares to biological learning, is looking at subcellular, maybe long time scale processes that occur in order to support synaptic changes for
Ah,
learning.
okay. Yeah.
So yeah,
So things that's going on inside synapses, maybe some like upregulation, downregulation
yeah, inside
of
synapses, and also maybe just things that are tracking kind of overall activity levels of
mm.
the cell, to
Mm.
have a sense of how active the cell has been recently, and
Mm.
relate that to outcomes of the animal's behavior, to say the activity of this cell is associated with good things happening or not.
Mm.
So
Mm.
that kind of thing, and yeah, being able to integrate different sources of information to do a calculation about how the synapse should change. All of
Mm.
that are things that could be happening subcellularly, and it would kind of be helpful from an algorithmic perspective if they were. So
Yeah.
now computational neuroscientists of a certain variety are happy to look at what subcellular people have been studying for a
Mm.
while.
Yeah, because one field under there is also this. I'm a physicist by training, so this molecular dynamics simulations where they simulate sort of like how they can, for example, simulate represent ion channels in terms of like proteins going back and forth through the membrane and sort of model it at atomic level. So there are, and this is of course this is used a lot in sort of to study other, protein folding and stuff like that. So there's also this field working at that level. And I guess if you sort of look at the Hodgkin-Huxley model, they had these key expressions for how these openings and closing of the channels depend on voltage, and this was sort of something they fitted to data. But in principle, these should be able to be computable, maybe from molecular dynamics simulations,
Yeah,
at least in
that's true.
principle, right? So there are sort of there is a level below there that
Oh,
sort of most we typically don't go into
yeah,
as
you can
computation
keep
neuroscience.
going for a really
Yeah,
long time.
that's true. Okay, so now we have sort of talked about the biophysical aspects, and this could have been about any body part, right? Whether it's brain or any other body part. I mean, it's the biophysics. We haven't really talked about coding and information processing, but that I guess there was this. That's the start of that avenue or that branch of computational neuroscience was really this paper by McCulloch and Pitts in '43. So what was that paper about?
Yeah, so that paper kind of demonstrated a way in which you could use what was known about neurons at the time, kind of their basic properties, that they fire an action potential and they have connections between each other. And some of those connections are excitatory, meaning that they make the other neuron more likely to fire, and some are inhibitory, meaning they stop the firing of the neuron. Using those basic facts, you could string neurons together in a way that they could implement logical functions.
Mm,
So the idea that a neuron is either firing or not, that can be related to Boolean binary logic, the idea that a statement is true or false. And then the connections between the neurons can implement these logical operators. So if you had a neuron that got two inputs from two different neurons, and it needed both of its inputs to be firing for it to fire, then that
mm,
neuron is implementing or the and function. It's saying I need my first input and my second input to be true, to be on, for me to be on, for me to represent true. And so through that, you could string together, infinite numbers of these to create more and more complex logical statements. And if you can do that, if you can make neurons do logic, then you're opening the door to explaining a
mm,
lot of the things that the brain can do about logic
mm,
and reasoning and all of that. So
mm,
that was their major contribution in terms of taking the physical details that were being studied by physiologists and biologists and giving them some kind of purpose in a larger computational system to actually
mm,
explain the outcome of the brain, more
mm,
explaining the mind using these physical details.
mm. And McCulloch was this physiologist, right, and working in Chicago. But Pitts was this very interesting character. Like, so he was also this child prodigy who sort of grew up in really difficult circumstances. And okay, I get it. I think it's a fascinating story. Maybe the most interesting person in neuroscience that I've come across.
Yeah, he was he grew up in Detroit, but he ran away from home when he was a teenager to actually go to the University of Chicago, not to attend it as a student, but just to like go hang out,
Mm,
because Bertrand Russell was visiting there and he wanted to hear his lectures and stuff. So he just kind
mm.
of hung out at
So
the
Bertrand Russell
university.
was this really famous mathematician, right?
Yeah, that
And philosopher. Mm,
yeah, who had a lot to say about logic and mathematics and all of that.
mm,
And so yeah, so Pitts just kind of hung out there. He was homeless for a bit, but
mm,
he just wanted to learn more about these things that he had read about as a kid, and clearly just had some innate ability to grasp these very complex, abstract, logical, mathematical ideas. And so yeah, he never really had a formal education or certification or anything like that, but he ended up functioning, as a scientist, as a
mm.
professor kind of person in the
Yeah.
field.
Huh.
But he also, for a variety of reasons, kind of struggled towards the end of his life and died rather young,
Mm.
because of some of the difficulties in his own life and in the science
Well,
that he was doing.
huh. And also, for those of you who have seen Good Will Hunting, for instance, like a movie from some decades ago with Matt Damon playing this prodigy that sneaks in, sort of like sort of correcting, well, sneaks in his like mathematical notes onto the professor's desks and sort of gets discovered that way. And that was a little bit similar to what happened to Pitts, right?
Hmm.
But
Yeah.
it wasn't really this McCulloch Pitts paper didn't make a big splash in neuroscience.
Yeah, the
Oh.
neuroscientists and the biologists and physiologists of the time, they didn't really know what to make of it because it didn't really explain their experimental data in any specific way. It didn't address questions that they had amongst themselves. It didn't tell them what they should look for in future experiments. It was just kind of making this claim that look, you can do logic with neurons, and
Mm.
they were like, okay,
then
but no
then
one was
what?
trying to
Yeah.
connect
No.
neurons to computation or logic at the time. So it
Yeah.
was a solution to a problem that didn't exist. And
Mm.
also, it was hard to read. Like the paper is not very clearly written,
Mm.
not
Mm.
even in retrospect. The notation of the
Mm.
paper and style didn't catch on.
Ah.
So,
Ah.
yeah, it wasn't taken up by the neuroscientists in
Mm.
any
Mm. And
enthusiastic way.
it was difficult to do. It didn't really. It wasn't any specific experiments that you could do to test it, maybe, or
Yeah,
at least not with
not
that. Yeah.
so clearly.
Mm. But it was sort of a starting point of another community, which is also obviously very important for the computational neuroscience community, the AI community. So how was that? So that was like this 56 conference or workshop somewhere in Dartmouth or something.
Yeah, so that's kind of the starting point of AI properly. But even before that, there were these Macy Foundation conferences or workshops where people that were kind of swirling around this topic area, like McCulloch and Pitts and John von Neumann and all of them, kind of in this realm of cybernetics or whatever it was called at the time. Um, that were really planting the seeds for all of that. And so through
Hmm.
that vein, McCulloch and Pitts had influence on this very burgeoning, very young field that, yeah, eventually got the label of artificial intelligence. But it was really in its starting point at this time.
And then because one thing that is sort of not so different, or at least had this maybe this logic idea, was this perceptron that with Rosenblatt. So what was that about?
Um, yeah, the perceptron came in like 1958. And it is also, an artificial neural network in the sense that it
Mm.
uses neuron-like elements, like the McCulloch and Pitts system does, to implement computations. But it has a lot of differences. I mean, one, it was like the Rosenblatt actually built a physical machine to implement it, to kind of simulate it. Whereas McCulloch and Pitts, they were just doing this kind of mathematical logical exercise in a paper. But the perceptron also is a bit different. It still has as its basic construction neurons that send connections to each other, and those connections are kind of the heart of the computation that are being done. But the perceptron doesn't explicitly implement these logical functions that McCulloch and Pitts identified as being possible in
Mm.
these networks. Instead, it learns to do kind of whatever computation it's trained to do by having a learning algorithm that changes the strength of the connections between these artificial neurons based on how well the network is performing on the task, based on if it's
Mm.
getting things correct or incorrect when it's asked to like classify some sort of input. So
Mm.
in that way, it has more utility, it has more functionality, especially applied to the real world, than what McCulloch and Pitts were doing, because it can actually be run and simulated and actually be trained to solve more complex and real problems.
Mm. And then Rosenblatt made all these grandiose claims and what he could do, and then there was like this story with the book of Minsky and Papered, who sort of like wrote about the perceptron, and then ended up sort of almost like making what's called the first nuclear winter. Not nuclear winter. The well,
That would be better.
what was the first neural network winter? Yeah. So
Less
it could have
bad.
been worse. It could have been a nuclear winter.
That
So
would be quite a book if it could create
yeah,
that.
yeah. So anyway, but so because it sort of like showed that you need this like you need a single layer network could do all these things. So that was a wrong. But that's like a different story. But what was interesting for me to learn by reading a book was that you actually found something like the perceptron in the brain, in the cerebellum.
Yeah, in the cerebellum, there's kind of it. the cerebellum has, compared to the cortex, has a somewhat neater organization to it, just looking at the cell structure by eye. A few different scientists were kind of came up with this idea that the mechanism of the cerebellum, kind of what was happening in the cerebellum with its different cell types, could be related to how a perceptron works. And so
Mm,
there's cell types that represent the output of the cerebellum that can go on to control behavior, the Purkinje
mm,
cells. And then there's cell types that represent the inputs, like sensory inputs. And so
mm,
basically, you have a system where sensory inputs are being transferred to behavioral outputs, and then there's a third set of cells that come in and signal error. So to say that, something about the behavior was not what it should be. And so with
mm.
those three components, you can have a learning algorithm that can update the connections from the sensory inputs to the behavioral outputs based on error. And that's kind of the whole heart of the perceptron learning
Yeah,
algorithm. And so that was found to be happening essentially in the
yeah,
cerebellum. And that's how the cerebellum can support error-based learning.
yeah. Because I mean, you have this Mars three levels of modeling, and so what are those again? So you can that's one.
So there's the computational level, which is kind of the
Mm,
goal of the system. Like, what is it
mm,
trying to do? So in this case, it could be error-based learning. Then there's the algorithm that the
mm,
circuit implements, and so in that case, it could be the specifics of the perceptron learning algorithm, how the weights should update in response to different types of errors. And then
mm,
there's the implementation level, which is what physical parts actually
mm.
realize that. So you have these different cell types in the cerebellum that play the different parts
Yeah. So
needed
this
to do the
this
algorithm.
so this cerebellum example is maybe the first example of actually a system where it was which was understood at all these three levels. Is that correct to
Um,
say?
yeah, I'm not sure. I mean, it depends on, how
Uh,
strict you are. You could probably find other examples earlier, but
uh,
it certainly is a very nice example of
uh,
it.
yeah. No, I think it's sort of is. So uh, because I think at that time also, if you look back at the modeling they did, uh, I mean, because it was also some of these like associative networks and like memory networks that was like started in the work on that in the 60s. And they often had very simple units. They didn't have anything like Hodgkin Huxley. They had very simple neural units, and it was more about what the network structure can do, and maybe like some kind of simple synaptic plasticity. So it was like, yeah, I guess it was well, I guess it was also for practical reasons. It was difficult to do
Yeah, to
networks, so
simulate these big
yeah,
networks
wasn't
of
really.
detailed neurons is a more recent ability.
Yeah, yeah. So and then another aspect, of course, is this. If you go back to when you started recording spikes, and I guess Adrian was like this Englishman who was sort of like was doing measuring all this spikes, and I came up with the first suggestion for I guess what is called a neural code. So what is meant by a well, what is meant? What did he do, and what is meant by a neural code?
Yeah, so the idea of a neural code is kind of, you try to identify what about neural activity you actually think matters in terms of what's carrying information and what's driving behavior. And so you can think of, as we said, neurons, they're firing or they're not firing. They have a shape to their action potential and all of that. And so there could be information hidden in multiple different ways within a neuron's activity. It could be
Mm,
that the action potential, the actual shape of the voltage changes, will change depending on the stimulus that's coming into the neuron. It could be that the
mm,
number of action potentials changes. It could be that the time between action potentials changes as a way
mm,
of, encoding, so to speak, the information that's coming into the neuron. What
mm,
Adrian showed was that the action potential is constant. It's all or
mm.
none. If the cell gets enough
Huh.
input to fire, it will fire, but the action potential itself will not change based on the inputs that are coming to the cell. So that
Mm,
kind of rules the shape of the action potential out as something that can carry information, because if it responds the same way every time, then it has no
mm,
ability to signal what has come into the cell. And so, through Adrian's experiments, by kind of putting in sensory inputs to different sensory nerve types, he showed that it seemed more convincing that the information was being carried in the number of action potentials that a neuron emitted rather than in anything about the shape of the action potential itself.
Mm.
So he was kind of an early proponent of this rate coding theory, meaning that
Mm.
the firing rate of a neuron is
Yeah.
carrying information that is, relevant for the computations that are being
Mm.
performed.
And alternative codes that I mean has been suggested later is sort of like there's like the detailed, I mean the timing between each spike really matters, and like I guess correlation code or yeah, I
Yeah, when
think
you're
yeah. So
looking
that's
at
like mm,
Yeah, when you start looking at multiple neurons, then you can also get kind of more complex codes about timing
uh,
between different neurons and all of
yeah,
that. So yeah, it's really in some ways it's open ended as to what you want to define or try to
mm,
test as coding in the brain, because there are many dynamic parts to neurons and
mm,
neural activity, and you can
mm,
argue that any one of them is encoding
yeah.
information to some extent.
So encoding that can be summarized saying that's essentially going from, say, for example, a stimulus to a pattern of spikes in a set of cells, a set of neurons. And so that's sort of like the encoding. And then, because lots of computational neuroscientists also works on decoding. So what's the job there?
Yeah, so that is more fraught as a topic, because you know, if you're thinking about this kind of coding as just a means of communicating information, of conveying information,
Mm,
then the decoding process should just kind of be a reverse of the encoding process. You go from
mm,
some input
mm,
stimulus into a series of spikes of a neuron, and then there's something on the other end that takes those spikes and reads them out and turns them back into an input
like a
stimulus, into the same
homunculus
stimulus,
or
which is
oh, yeah.
not what we want our brains to do.
No,
We don't want them to
no,
just recreate the inputs that we give to them.
no. We don't need
So,
that, right? It's true.
yeah.
Yeah.
So, really, there, in some sense, there kind of isn't that, you know, very basic decoding that happens in the brain, because there's
Mm,
transformation of information that happens in the brain, and there's computation that happens, and you're supposed to take in inputs and do something with them to create
mm,
different outputs.
yeah,
So what is decoding in the brain is, it's a far broader
mm,
question, but it still relies on the fact that information is encoded in neural
yeah.
activity in some form, such that other
Mm,
neurons can get input from those neurons, and that can, create something
mm,
useful
yeah.
and behavior.
Because I guess a general problem, if you see some kind of neural representation in there, which in principle contains information about something about, I mean, about the outside world, it doesn't mean that the nervous system is able to extract this information, right? That's a
Yeah, and
different
this
thing.
is something people fight about a lot now.
Okay.
In papers, if all you did was record neural activity and showed
Uh,
that you, as the experimenter, could use some sort of algorithm to read out the information, and you can say
uh,
the information is kind of in that neural activity,
uh,
is that, what is that saying about the brain itself?
uh,
It's saying the information is present in that neural
uh,
activity. It's not saying that there are neurons in the brain that are reading out that information from that neural activity.
uh. I guess I mean you also have this now this question about sort of like the like other things than the spikes, but also like the local field potential or the EEG, and nobody would say that the EEG itself is used by the rest of the brain, right? So even though
There
it can
are
stay,
people who would say that the local field potential is
yeah,
carrying
that's true.
information
Yeah,
to the other
so
neurons. Yeah. So
that could
that's
be
not
that could be.
technically like a neural signal, because it's not directly coming from the neuron, but it's kind of a
Mm,
diffuse signal of the
yeah,
activity of a bunch of neurons that
yeah,
that could be
yeah.
carrying
So that's
information.
true. That could be it. So that they're the I guess the jury is still out. We don't really know.
Yeah. Yeah.
To what extent. But so that's good. But then you had like this other theory that came sort of like for quantifying information, which came from Claude Shannon. That was all like just well, I guess it was just published just after the war or something.
Yeah, yeah. So he developed this kind of formalization of the concept of information, because it's something that, like, we've been using and neuroscientists have used for a long time, but without actually having a formal definition, it's hard to study it in the
Mm,
brain or anywhere else. But his motivation was coming from thinking about communication and thinking about things like communication during the war, where how can you make sure that you're sending information that's reliable but can also be sent quickly, and that kind of thing. And so his notion of information depends on actually kind of already knowing what the code is. You have to be able to say what a symbol in this code is, and then if you can say that, then you can do a simple calculation to say this is how much information is carried in these symbols in this code. But it doesn't solve the problem of not knowing what the code of the brain is. It's just a way of talking about information, quantifying information, if you have a guess about what the code is.
Because I think it was like when it came, it was like a formalism you could apply, so you could compute things like bits per second or bits per spike and stuff like that. So, but it was I think even Shannon himself got a little bit worried about the over application of his ideas.
Yeah, he wrote this short article a few years after he published this definition of information, and just saying like it's being applied too much. It's being people are attributing to this concept more than it's capable of providing, and that people should be cautious. Because it was, again, it's based mostly on the concept of communication, and so it's just about sending a signal and then being able to recover the signal that was sent. And as we were saying, in the brain, you're doing more than just sending a signal. You're trying to transform it and combine it with other things and create useful output from it. And none of that is inherent to the original definition of information from Shannon.
Yes, one way to maybe to get to closer to, well, have ideas about like the codes that are used is to have some kind of principle. Like one principle is this idea of efficient coding that came, I guess, late 50s or something. So what was that all about?
Yeah, so that comes from Horace Barlow for the most part, and that the idea there is that basically the brain would not be redundant. The brain wouldn't be sending signals that are not helpful or necessary. And so if something in the environment is redundant, if something in the environment is happening over and over, the brain would stop sending a signal associated to that. And there are kind of other ways in which you can reduce redundancy in a code, which are, it's based on the statistics of the information that's coming in and being transmitted, how you decide what's redundant and what makes a code efficient as a result of that. So that was his idea, that because of, the metabolic cost of sending spikes and all of that, that the brain is going to use an efficient code. And if we keep that in mind, we'll be able to understand some of the peculiarities of how neurons respond to things, or really how neurons don't respond to things as well.
Cool. So actually, we're going to have an upcoming podcast about the neural code with Arvind Kumar, which will be soon at the podcast close to home. So yeah. So, but I think now if you go sort of to starting now, we have talked about these principles and these ideas, and I guess the system that was like, which was really has gotten a lot of attention, I guess, is vision. Vision has a lot of, I mean, one thing, we are very visual animals, and so that, I guess it was this, I think you mentioned this Jerome Letwin guy, also just after the war. So what did he do?
Yeah, so he was part of a study on frogs that spoke to this idea of the visual system as being a hierarchy of feature extractors. So if you kind of maybe just had like the most basic sense of how vision could work, you might think that if you're trying to recognize something, like a coffee mug, that you just have stored in your brain some template of what a coffee mug is, and information comes in, and you compare it to that template, and that's how you decide. That was kind of a very basic theory that was being tossed around, but it has a lot of practical challenges because most things aren't so easily describable that a single template would explain all of them. And also, if you're really thinking about a template at kind of the pixel or retina level, just changes in lighting, changes in angle, and all of that you're going to need separate templates for the same object under different conditions. So it's just not really a feasible way to solve vision. But the idea of using a hierarchy of feature extractors can build in some robustness. And so the project that Letwin was a part of was recording from the retinal cells in a frog and seeing the different types of light patterns that they respond to. And so they found a few different cell types in the frog retina, that they weren't just kind of conveying verbatim the light hit this part of the retina and that goes on to the brain. They were doing some computations themselves. And so one of the most striking computations was that, you'd have neurons that would respond if a small dark dot kind of moved quickly across the visual field. And the natural conclusion or assumption there is that these can help the frog detect bugs that they're trying to catch. And so having part of that computation built in, even at the earliest layers of the visual system, means that the visual system isn't just, the retina isn't just taking in information and doing a single calculation later. It's kind of pulling out the relevant bits throughout the hierarchy.
But already this idea is quite different from the idea of McCulloch and Pitts, right? Because these neurons were doing much more than just sort of doing this binary operation, right? It was not like Boolean things they were doing. So I guess that was also quite an argument against that idea.
Yeah, and it's points more in the perceptron direction, in the sense that it's kind of just messier and more spread out. It's, you have things are getting done where they need to get done to achieve the desired behavior, and you can't put a clean label on each of the parts usually.
And then this was taken on and generalized or tested out in cats, particularly, both in, I guess first retina and also LGN, and then the visual cortex with Jubilan and Wiesel. So of course, their work is quite well known. So what did they do?
So they discovered that, as you said, at that time, people knew that there were cells in the retina and cells in the lateral geniculate nucleus in the thalamus that would respond to kind of points of contrast, like a dark point surrounded by a lot of light, or the reverse. And so they kind of went into the next stage of visual processing, which is the visual cortex at the back of the brain, and initially started looking for cells that also responded to those kind of contrast points of light. But what they discovered was that that's not what those cells responded to. They actually responded to oriented lines, kind of more like bars of light, sometimes that are moving, and that the neurons had very specific responses. They only responded to a particular orientation at a particular location. And particularly for what they called simple cells, it really had to be in the exact precise location. For complex cells, it could be in one of a few locations that were near each other. But on the whole, they all had these very particular orientation preferences. And so through that, they were able to kind of guess about this hierarchical model whereby the LGN cells have respond to these points of light, but if you align a bunch of points in a row, you get a line. And so the orientation of those points leads to the orientation preference of the visual cortical cells. And then also the spatial preferences of the simple cells. If you give a few simple cells as input to a complex cell, then it responds to the line in multiple different locations, whereas the simple cells are responding just to their one location.
So this was a really cool idea, and then of course they wanted to see, does this work at the higher visual areas? And there it wasn't so clean cut, right?
Yeah, they kind of tried to keep. They were looking for like hyper complex cells and kind of to keep going, but it did start to fall apart. And there was really for many decades following Hubel and Wiesel, there was this attempt in a lot of studies in vision and other mostly sensory areas, that the goal is to kind of categorize cells, to record from a bunch of cells and then be able to say, these are the simple cells and the complex cells, or these are the curve detectors and these are the star detectors, or something like that. And that only had medium success in terms of actually being able to explain things in a satisfying way, because the cells weren't that categorizable. They didn't form these kind of neat categories, especially once you got past the primary visual cortex. So Hubel and Wiesel then just started studying development of the visual system, and particularly of how this orientation preference develops in V1.
And then in Japan, there was this Fukushima guy. So he took this idea and he was like a computational neuroscientist, and he took this idea to construct a network. He called it, what's called neocognitron or something, was it?
Mm-hmm. Yeah.
Yeah.
Yeah, he heard about Hubel and Wiesel's work through fellow employees at the radio station that he was working at as an engineer. And he heard about it and thought, okay, this is kind of the algorithm for how vision is done in the brain. You have these cells that respond to oriented edges, and then you have these other cells that get input from those, and have some more spatial invariance as a result. And so he built this very early computer vision system that was based on the principles of Hubel and Wiesel, but he kind of extended it beyond by kind of replicating this pattern of, you have these simple like cells that look for patterns, very simple patterns in the image or in the inputs that they get, and then you have the complex cells that get more spatially invariant, and then you repeat that pattern of looking for patterns and making it spatially invariant, and looking for patterns and making it spatially invariant, until you can go, you can have a whole system that takes in an image and puts out a label of what's in that image. Now, his images were very simple. They were like binary, like black and white images of digits, so just like very simple lines and stuff. But the concept was there, that you could build this hierarchy of feature extraction, essentially.
Mm. And I mean, another lot. I mean, at that time, I mean, in the 70s, there were also people working on this, making these firing rate models. So that was like, so far we talked about like single neuron models and biophysically detailed or more simplified leaky interrate to fire neurons, but still neurons that fire spikes. But then you have these firing rate models, and they were, I guess, it was like Wilson and Cowan was like two early pioneers there who made this, where you sort of model the probability for firing or firing, like firing rates of populations or something. So they became they were used in studying the visual system also, right? And I think one really cool recent example, which you highlight in the book, is these ring networks. I mean, of this head direction cells in the fly
Yeah.
visual system. So that's because there they use, yeah. So I think that's one of the examples of, well, if you can tell, remember those.
So the fire rate models, as you say, they're not the same as like leaky integrated fire or Hodgkin-Huxley. The neurons in those models are represented by a continuous variable that is supposed to represent something like firing rate, which is consistent with the general belief in rate coding in the brain, right? If you think the firing rate is what matters, then you can just have a model that captures that, and you don't have to capture each individual spike. But yeah, those models have been used, throughout a lot of different areas of interest in computational neuroscience, because again, by making it a little bit simpler, you can study more complex populations of neurons if you make each individual neuron a little bit simpler to model, and you can apply like a lot of linear algebra and dynamical systems analysis to be able to make some nice statements about how these populations of neurons behave. And one of the examples where that has really been kind of quite elegant is in the head direction system. It exists in flies and mice, and I assume most animals need to know which direction their head is facing. But the idea there is that you have a set of neurons that are connected to each other in a spatially dependent way. So there's like nearby neurons that are connected to each other, and then there's farther away neurons that don't connect to the original neuron but connect to their neighbors. And if you have this in the shape of a ring, you can think of different locations on this ring of neurons as representing different locations, different directions for the head to face, because it can go in 360 degrees. And so the firing rate pattern of the neurons in this ring of neurons, which neurons are firing, indicates which direction the head is facing. These ring networks, if they're built exactly right, can kind of have memory. So if there's a way that the head is facing, and a lot of times animals get this cue based on kind of visual inputs or other things in the environment, they know which direction their head is facing. Even if you turn off all the lights, they still have a memory of which direction their head is facing, where they're facing. And that works because the neurons in this network, their connections are such that it's known as, there's an attractor state, meaning that the neurons will sustain their own activity even in the absence of inputs. And that's really important for memory more broadly. And it comes out of, as I said, kind of this notion of an attractor is you can study it really elegantly with dynamical systems theory. It's a very core concept there. But yeah, in the heading direction system, you can also then give them the model inputs that update the direction of the head in the representation, and it can just really function very well with a relatively simple and elegant system. So computational neuroscientists like it.
Yeah.
And then it was
Also,
discovered,
yeah,
in a very, again,
uh,
elegant and beautiful finding in
yeah.
the
So
fly
that's
brain, that these neurons, which we think of as a ring network as an abstraction, actually
yeah,
formed the shape of a
yeah.
ring in
So
the fly brain.
that's what we call textbook material,
Yes,
right? Yeah.
exactly.
Okay.
The textbooks were
As,
being rewritten as soon as that
yeah,
paper came out, I'm sure.
exactly. So I think it's something about these simple models, conceptual simple models that can sort of interpretation is clear. And so I just want, for completeness, I guess so far we have talked about what we could call mechanistic models, which sort of like all of this has been about mechanistic models, at least the biophysical models, and also these like the ring networks. But you also have these descriptive models, more like I mean, for example, the Gabor function, models which are just used to capture like experimental data. I guess that's, I mean, often you see this, like a difference of Gaussian's model, which are used for the retina and also the LGN. And then we have these Gabor functions that is just sort of kept in the, for the simple cells, just telling sort of giving in, it's a way to capture information capture and store the information about sort of like the response of a cell to different kinds of visual stimuli. And we're not going to talk so much about that, but that's also sort of obviously a part of computational neuroscience.
Yeah.
Okay, let's go. Mm.
Yeah, and the descriptive models actually interplay with these mechanistic models, I think, quite nicely, because usually, at least my perspective on it, is that you need the data to be described in a nice compact form when you're trying to work on building a mechanistic model, or at least it certainly
Mm.
helps if you have some sense of, if you don't have to deal with kind of the messy data itself, but you have some abstracted description of it. So kind of it's already put in mathematical terms and it's already simplified in a way so that your target of what the mechanistic model is trying to capture is clearer. I think that's one way in which they can interact. They don't have to. Like sometimes descriptive models are just on their own, and that's fine. But I certainly see a connection between the descriptive and the mechanistic in that way.
Mm. One thing we also, I mean, there's also this, of course, data analysis. That's a whole, I mean, lots of computational neuroscientists work on data analysis. And so there's all this thing about, which I guess is in I mean, these descriptive models are a kind of data analysis, right? You sort of try to sort of fit the data, describe the data in terms of these like receptive field models.
Yeah.
But then you also have all these dimensionality reduction techniques, which
Yeah.
are,
Yeah, that's becoming increasingly popular as the recording methodology makes it such that more and more neurons are being recorded at the same time. Back when Hugh, Will, and Weasel and those kinds of people were working, they could only record from one neuron at a time, and so it made sense that they were trying to categorize these neurons and describe these neurons in this very individual way about what is this neuron doing. But when you're recording from 1,000 neurons at a time, you're not going to go through one by one probably and try to give them all little
so,
names or anything like that.
no,
And so the question is, what do you do with them? And so these
yeah,
dimensionality reduction techniques are becoming increasingly crucial for how people visualize and try to make sense of a big pile of neurons that they recorded by taking those neurons and combining them in ways that you can make this simplified plot and deal with, instead of 1,000 neurons, you can deal with 3 dimensions that represent a lot of the variability that's happening in the neural activity.
mm. And then you use this like PCA, principal components analysis. It's sort of like and then you do this. I guess you make this neural manifolds. Is that sort of like and you
Yeah,
sort of plot
that
how these components move around in.
Yeah, that's another
Yeah.
one that there's a lot of debate over how those concepts should be used and the utility that they have.
Mm. Yeah.
But certainly, yeah, people are really just trying to come up with new concepts in a way for how you understand large populations
Mm.
of neural activity. What should we be trying to pull out of them, and how should we
Mm.
describe the things that we find and relate them to things like behavior or whatever other
Yeah.
useful computations you think are happening?
Yeah. Because now, I mean, with this new, I mean, if you just look at electrophysiology with all these like neuropixel probes that you can put in many at the same time, you can measure from like thousands of neurons, and with two-phone calcium imaging, it's even more.
Hmm.
So what should you do with this data? You really need some techniques for handling them.
Yeah.
So this is sort of an obvious need. So, but now, I mean, so far we've talked about computations, I guess, like, but we haven't. Another important aspect of the brain is memory, and this idea of Hebbian learning. That's
Mhm.
like a key thing. Can you just like give a short intro to the Hebbian learning before we move on to the Hopfield model? I don't. Mm.
Yeah, so Hebbian learning is named after Donald Hebb, and it was kind of a speculative thing that he put forth to say that there's neurons that are active together are going to form stronger associations, and the way people say it now is like neurons that fire together wire together. And so, but it's a simple idea, but it has been explored and can lead to a lot of useful things when applied at a broad population level. But yeah, it's just the idea that if especially there's versions of it that are more temporally dependent, but if one neuron spikes and then the neuron it connects to spikes after it, then it seems like that first neuron caused that second neuron to spike. And Hebbian learning would say that those that connection between those neurons should grow even stronger, so
Mm.
that this first neuron will continue to cause the second neuron to spike. So
Mhm.
it's kind of simple in that way. It doesn't actually involve any feedback about what happened after those neurons spiked and was it a
Mhm.
good thing or a bad thing or anything like that. The most basic
Mhm.
form of Hebbian learning doesn't. It just says that things that happened temporally close in time should create a greater association.
Mhm. Because it's spike timing dependent plasticity which was I guess was discovered in the late 1990s or something. That is so that's sort of like a Hebbian. Yeah, that's really is this Hebbian ID at the
Yeah.
single neural level.
Yeah, Heb's original conception, I don't think spoke of temporal
No.
order
Hmm.
necessarily,
Yeah.
but yeah, just the idea that things that were, firing together, associations should be formed between things that are occurring
Hmm.
at the same time.
Hmm. And then you have the Hopfield model, which is sort of like the yeah. This is a really nice and popular model, which which everybody likes. In a sense, it's a beautiful model. It sort of is. Yeah, it gives a lot of insights, at least qualitative, with rather simple mathematics and elegant mathematics.
Yeah, the Hopfield model really
Mm.
represents I mean, you could maybe divide kind of the history of computational neuroscience into a first wave that was really like the electrical circuit kind of models that were more on the biophysical level. And then in the 80s, you get this influx of physicists who want to study the brain. And
Mm.
Hopfield himself and his model, the Hopfield network, really
That's
represent
right.
that era
Yeah.
where from what I've heard from these physicists, it's that they found physics to like already be solved. Like too many
Well,
of the mysteries were
that's
not
true.
mysteries anymore, and so they were turning to the brain where
Yeah.
things were a
And I
lot
guess
wider and
many
more
of them came
open.
from statistical physics also,
Yes,
because that's
statistical
like the
physics.
branch with many units.
Yes,
Yeah,
exactly. It
it's
was
like cooperative properties, so it sort of has like
yeah.
these spin
So
glasses and stuff. So, yeah.
in that way well suited for the brain. You can see why they
Mm.
saw that connection.
Yeah.
Yeah, that like where we were studying individual atoms and how they create large scale behavior from their interactions, and the brain is a
Mm.
bunch of individual neurons that interact and create large scale behavior. And
Mm.
so that's kind of what the Hopfield model is based on, the idea that you have these individual neurons, and by changing the connection strengths between them you can create these large scale behaviors that Hopfield associated with memory states. So you
Mm.
can create a network that can store memories and recall memories
Mm.
based solely on the interactions between the individual neurons.
And it also had this like associative thing, or that you can sort of complete memories. That if you
Yeah,
have
you
like
can
a
put
yeah,
in a partial pattern of a memory
mm.
state. So it kind of just turns some of the right neurons on or off.
Yeah.
And then the connections between the neurons will reinstate the full memory pattern, the full
Mm.
activity pattern that
Mm.
represents that memory.
Actually, back in the days when I used to do semiconductor physics, well, took a PhD in semiconductor physics, and then when I switched to neuroscience, I went to this meeting in Sweden where Hopfield was. I was really new in this field, but I knew about the Hopfield model. So I asked him sort of what was his role, what did he think was the role of his Hopfield model? What role would it have in the end? And then he said it's going to might be a metaphor for how the brain works or how memory works, and that was sort
Okay.
of yeah. But at least it's certainly something about models which are not certainly not well correct in the details in any way, but still very useful
Yeah.
because it
And
gives us a way to think about. think about it.
Yeah, there was pushback from neuroscientists of the time of like, this isn't
Yeah.
neuroscience.
No.
But then, that approach has carried on in
Mm,
various forms. The more abstract approach, because it does seem to still highlight useful ideas about how the brain could be working, even though it's not
mm.
like these biophysically detailed models that came before.
Yeah, no, because it's also that like with these biophysically detailed models, at least these large network models, you can sort of do simulations that get out results which are hard to get your head wrap your head around, right? So at least with these simpler models has this advantage that you can sort of understand the consequences and sort of
Yeah.
get some key ideas out maybe. So but these attractor models, that's also, I think you mentioned it also, that this had been also for a working memory, that some of these attractor models has been used.
Yeah, so the attractor model is the Hopfield network is an attractor model, and it can be used for thinking about long term memories as well, like this idea of putting in a partial bit and recovering it.
Mm,
But then, yeah, attractors are also helpful for working memory because it's just a description of kind of the dynamical properties of the model that. When there's a certain state that it can be in, where
mm,
the neurons will sustain their own firing, and even if if the attractor is built the right way, even if you kind of perturb the system a little bit, it'll still go back to that state, so it'll hold on
mm,
to that memory even if some inputs are coming in. If you perturb the
mm,
system too much, it'll get knocked out of that state, but
mm.
it has some basic properties that would help hold on to something, which is
Mm.
what you need in working memory.
Yeah, and this is like the ring network model that you
Yeah,
talk about is
exactly.
an example of that, right? So
Yeah.
so that's sort of like obviously this you need some kind of memory both at like different time scales in the brain, and this like attractors isn't attractive, I guess. Attractor is an attractive alternative there. I mean, when I look at sort of, I mean, I think if you sort of if I wanted to send like in text message to a foreign civilization about what we have learned about the brain, I would say, well, I think we understand a single neuron fairly okay, but we are really with networks we are sort of really struggling. But I guess it's not that many specific concrete insights that has come from, I think, network studies, at least, or like by like mechanistic network studies. But I think this idea of a balanced excitation and inhibition is one of them. So that's sort of like something we can sort of put in our belt and be proud about. So what is the problem there? Or what?
Yeah,
Yeah.
so kind of one way into understanding this notion of balance is to think about the variability of response in neurons. If you
Mhm.
give this a neuron in a network the same input over and over, like an external current or something like that, it won't respond exactly the same way each time. And so that was a mystery for a while as to why neurons weren't behaving in a
Mhm.
kind of clean, deterministic way, and why they had this noise in them. And the solution, or the kind of mechanism that's been found to explain why they're a bit noisy and a bit erratic in a way, is this fact that neurons get a lot of inputs, and those inputs can be excitatory or inhibitory, and they're basically getting a lot of both of those. So at any given
Mhm.
moment, they're being strongly driven to fire, but also strongly shut down by the inhibition. And when you
Mhm.
have those like two heavy things leaning against each other, if there's any
Mhm.
slight imbalance, kind of the whole
Huh.
system falls over, but it can go in different
Mhm.
directions. And so that tight balance between excitation and inhibition puts networks of neurons in this kind of precarious state, where they're
Mhm.
very subject to small fluctuations in inputs can
Mhm.
cause big changes in the output.
Huh.
And so this is achieved through processes that make it such that neurons get a roughly equal amount of excitation and inhibition coming into them at
Hmm.
any time.
But it's sort of like driving you with your gas pedal and the brake pedal in at the same time. So it sounds a bit too seems a bit wasteful a
Yeah,
priori. So why
that's
why
true.
what's the advantage of this?
Yeah, I guess it goes against the efficient coding in some way. If we're just like creating all
Wasteful
this
code.
input just to cancel it out. But
Uh,
one thing that people have found is that it does lead to faster responses in neurons. So if it was like
mm,
neurons were completely off and not getting any input, and when they were supposed to fire, then you just start getting them input, and then you have to wait for them to kind of integrate and then fire and everything like that.
mm,
That could make it slower to respond. Whereas if you have the excitation like just at the ready and you just need to kind of let the inhibition go down or push the excitation a
mm.
little bit more to overcome the inhibition,
Exactly.
um, then you can get this really fast
So it's like
response.
shooting across a traffic light when it comes to rules, right?
Yeah.
If you want to
Yeah.
impress the neighbor.
Yeah. It's just
Yeah,
always at the ready.
yeah. No, that's a sort of like a cool idea. I guess that was developed in the understood in the 90s.
Yeah. This was part of the
Yeah,
same wave of the physicists
yeah,
coming into neuroscience
yeah,
using their,
yeah,
their physics models.
yeah. And then you had like I think like one paper I really like, which is this Brunel paper on the about the paper by Nicolas Brunel on this phase diagram,
Hmm.
where you can sort of see how you with different you have this very simple network with integrated fire neurons, both excitatory and inhibitory, and by changing the weights a little bit, you can get all these kind of different firing patterns. Not only this very noisy pattern, but also the regular oscillations and so on. So I guess we are sort of we understand some generic properties of networks. It's just very difficult to translate it to understanding one particular well biological network. I guess we're not quite at least that much. That's much harder. So I mean, one piece one part of computation neuroscience which I never worked on myself is this with this but is important, well, both because it's a important problem in neuroscience, but also because it has so and many it's so linked to applications in AI, is this thing with reinforcement learning. So can it, because that's also one of the success stories maybe of computation neuroscience, where you have these explanations at Mars 3 levels of understanding.
Yeah, so reinforcement learning refers kind of generally to the idea of learning from rewards rather than learning from particular feedback. So it would be like
Mhm.
if you took a test and instead of actually getting the graded test back where you knew what you did right or wrong, you just got the grade and you just knew like you overall did a good job or a bad job.
Mhm.
So that's pretty hard because you don't know what contributed to the reward you got.
Oh,
If you got what reward, you don't
exactly.
know what contributed to not getting the reward.
That's
So
true.
it's a really difficult problem. But animals can clearly do it. There's clearly a lot of reward based learning that goes on
Uh,
in humans, but in animal behavior
mm,
as well. And so, yeah, the story of reinforcement learning really spans a lot of different influences. It's like people who were simply studying behavior, people who study neurons, people who study artificial intelligence. Everyone was kind of adding their angle to it.
mm,
Um, but yeah, one of the kind of major so yeah, if we think about on Mars 3 levels, the computational level is to learn from rewards. It's to learn to do well
mm,
because of reward feedback. And that means learning what actions to take well in advance of when you'd actually get the reward.
mm,
And so that was a problem that was of interest to Richard Bellman, who was
mm,
working at the Rand Corporation, and he was an applied mathematician. And so he was trying to solve those problems for, kind of industrial application. Any business has to make decisions that will later result in reward or not reward.
mm,
And so he came up with this value function idea, which is this idea that you want to learn a value function, meaning you want to learn in any given state that you're in, is it a good state or a bad state? So you won't know what
mm,
reward you get yet, but you can have a sense of like, am I in a good state or a bad state? And this can happen it's kind of intuitive. Like if you play chess or something like that, someone could just plop you down and in front of a partially played chess game, and you'd be able to have a sense of like, ooh, this is not a good state for me to be in. This is like a
mm,
bad board state for me. I'm
mm,
probably not going to win this game. So you
mm,
have this sense of value, even though you're not getting the reward feedback. You don't know if you won or lost yet. You have a sense of like, from this position, how close am I to reward? How
mm.
well set up am I? And so the
Ah,
goal is to learn that value function. If you play chess a lot, you learn it because
mm,
humans are good at reinforcement learning to some extent. You
mm,
will learn that value function for these different states. And so Richard Bellman was trying to come up with, he kind of defined this concept of value function, that's what you're aiming for, because then then you can make the decisions well in advance of when you actually get the reward. You can make decisions as you play the chess game that get you to a better board state that has a high value where you feel like you will win. But you can make these decisions based on the value well in advance of when you get the reward. But then the question is, how do you actually learn this value function? And that's where some more influence from behavioral studies and that sort of thing also came in. Sutton and Bartow are also computer scientists / cognitive science. This whole realm of reinforcement learning is very mixed in terms of what kind of scientists these people are. But this idea that you can learn the value function through exploration, where the key thing that matters is that you make a prediction about what reward you'll get, and then you compare that to what you actually got. So the key computation is doing a reward prediction error calculation. And if you update your value function when you incorrectly predicted reward, then you will learn a value function that can be very helpful for getting reward in the future.
Yeah.
And then this has been borne out in a lot of various animal experiments as well.
Yeah, because the maybe the surprise, or at least for me surprising, is that you get this at the neural level, you get this dopamine rewards or whatever, like dopamine squirts. But it's not like you get the dopamine reward when you, well, you don't get dopamine as a reward. You get the dopamine when you to signal error, right?
Yeah. So
Error in the prediction.
Yeah, people kind of associate dopamine with reward or with pleasure or happiness or that kind of thing. But in fact, it is a signal, at least, the kind of current computational framework for understanding dopamine is that it's a signal of reward prediction error. But if you predicted that you weren't going to get a reward and then you did, that's a positive error. And so, that's how it becomes associated with something pleasurable, because you got a reward when you didn't think you were going to get one. But if you expect a reward, if it's like it's payday and you get your paycheck, there's no dopamine there. There's no even though it is a reward in some way, you got money, you got something you wanted, but you expected it, so you
Like
don't
this.
get any pleasure from that.
It's
And so
like
it's
this 50-year-olds who have everything, right? They don't they don't get any pleasant surprises anymore because they have
yeah.
everything.
You
Yeah.
need to be, your expectations need to be violated.
Yeah.
So it was found that that's a better description of what dopamine signals, is this kind of reward prediction
Yeah.
error idea. And that makes a lot of sense because dopamine also plays a role in gating synaptic plasticity, of gating changes in connections between neurons, which is how learning happens. So if dopamine is there, it means you made an error in your prediction, and therefore changes need to happen in the network so that you don't make that error again.
But where in the brain does this happen? It's not necessarily in the cortex, right?
It's all, so the dopamine neurons generally come from subcortical areas, but they really spread out to a lot of places. So I think
Okay.
it's happening
The
all
whole
around.
circuit, because it's also like the striatum. It's not like this one of these areas I just know the name and I barely know what they do.
Yeah, so there's dopamine neurons there, but I think they send their projections to a lot of places. Yeah.
I see. Yeah. So that's also one of the successes we can sort of brag about. I guess this idea that idea about this error signal came actually from theory in some sense first. Yeah. And then it was sort of like
kind of from computer science wanting to build systems that could solve this difficult problem.
That's cool. And then, we have this new way of doing computational neuroscience, which I think is the type of neuroscience you are interested in now, or computational neuroscience. You're interested using AI. So to sort of because the hard. When you try to fit sort of like say these large scale cortical models to experiments, it's very hard. You don't really have a learning rule. You have to sort of to try to fit experiments. It's not very easy to train or to fit these models to data. But with these AI models, you have these beautiful learning rules, right? So you can train the AI models, these convolutional neural networks and other architectures and that is quite successful that works quite well, right? So that is sort of so that's the advantage of that type of network. So because that's like a new branch of computational neuroscience.
Well, you could say it's new, or you could say it's revisiting of the perceptron, a revisiting of the time in the 80s where people were doing a similar kind of approach. But the the benefit there is, yeah, as you say, you can train these networks, but importantly, for the most part, people are not training them to capture neural data directly. They're training them to produce a behavior, like being able to classify images and that kind of thing.
Mm. Yeah.
And so in that way, it's a more behavior forward approach than I think actually a
Mm.
lot of computational neuroscience had been, and maybe
Mm.
even like kind of systems neuroscience more generally. It really matters that you're thinking about what behavior you think the network
Mm.
should be generating, and making sure that you're studying a model that can actually do that behavior, versus,
Mm.
you know, some of these like circuit models we're talking about. They're not meant to complete a full input output, you know, that the
Mm.
organism does. They're meant to replicate a very small part of the brain that's part of a larger circuit, and you make
Mm.
assumptions about how this small thing that you're modeling fits into the larger system in order to create behavior. But with these artificial neural networks that we have the data and compute to train to do full tasks, then you can really be studying a model that's actually doing the behavior of
Mm.
interest.
But then you often go in afterward in the trained network and try to make connections to, like, particularly in the visual system. I guess there's been some, like, when they sort of looked at this AI networks that's been trained to for image classification, and then when you look into them afterwards, then they have something that resembles the neural representation and receptive fields in the visual system of mammals, right?
Yeah, that's one of the cool things that comes out of it, and what kind of further helps verify that these are good models is that then, yeah, you can go inside and you can make comparisons between the artificial neural activity and the real neural
Mm.
activity in response to the same inputs. You know, you can literally
Mm.
show a human or a monkey an image and show the same image to the neural network and compare the neural responses.
Mm.
And yeah, in vision in particular, that's been quite successful on the whole, and people are interested in finding out what exactly about the neural network is helping
Mm.
it match the real neural activity. You know, is it about how you train it, the data you train it on, the architecture of the network, trying to figure out what makes the model the best match to the neural activity, so that you
Mm.
can say something about the
Mm.
visual system in the brain itself. And yeah, people have kind of deployed this in other sensory systems and other computations of the brain as well. So yeah, for the most part, the setup is you don't train on neural data. You're not training the model to predict the neural data, but you're
Mm.
training it to do a task, and then you're able to relate the artificial neural activity to the
Mm.
real neural activity, because they're both doing the same task, and hopefully you've given kind of the right architecture that you're mimicking how
Mm.
the brain is doing it.
Mm. I mean, your webpage, I got the impression that you are working on these things these days.
Yeah, that
So,
my research is mostly around building these artificial neural network
well,
models.
can you tell a little bit about it, or?
Yeah, sure.
Yeah.
So a big part of what I do is study attention,
Mm.
which is a big concept. It has a lot of different definitions, but I'm mostly focused on sensory attention. So just this idea, like if you're looking for your car keys, there are changes that happen in your visual system that make you better at finding them. Like kind of the
Mm.
neurons that respond to shiny things of a certain shape, they'll have their firing rate increased so that you can help that will help you detect the thing that you're looking for. And so I kind of use these models because they can actually do behavior. You can show them complex images and ask them what they see in the image. And then you can also add in mechanisms that mimic attention in
Mm.
the brain, so that you can study
Yeah.
exactly how attention changes behavior on complex visual tasks. I'm
Huh.
also studying auditory tasks now. So that's kind of the reason for using these models, is that for me to understand something like attention, the whole point of attention is that it enhances your performance at difficult tasks. And so I need the
Mm.
model to be able to perform difficult tasks. And then I also
Mm.
need a model that has something like neurons that correspond to different brain regions, so that I can put
Mm.
in the biological mechanisms that I think are at play. And
Huh.
these models have both of those. So it's really nice for me. I think it's a nice model system to study attention in particular
Mm.
in.
So, how do you include attention specifically in your models? How is
Yeah,
that done?
so the basic idea, you know, again, my goal, because so some people kind of work in this neuro AI space, and partly their
Mm.
goal is to take the biology and make AI better. But
Mm.
my goal is mainly to use these artificial neural networks to build models of the brain and just make them as biologically correct as
Mm.
possible.
Mm.
So I get I look at the literature, the neurophysiology of attention, how it changes neural activity, what kind of neurons that have their activity changed by attention, and all of that. And for the most part, it comes down to changing the gain of the neuron, changing kind of the slope of the input output function for certain neurons that represent the attended objects. And so there's a few different ways you can control that in these artificial neural networks, but the point is you kind of come in and you gain modulate parts of the network, and then you see how that impacts the performance of the network.
Cool. So, well, we approaching the end here, so I think we can sort of, I said, put our head up, put our hair down, and be a little bit more speculative. So it's fun to talk about sort of like this. I remember I read this book, Dreams of a Final Theory, which had to do with by Steven Weinberg. It had to do with particle physics.
Wow.
So what's your dream of a final? Do you have a dream of a final theory for the brain, or whatever? What would that mean?
See, it's funny because I think it is very common. I don't know about other fields, but certainly in neuroscience, people look to physics as like, this is how a science is meant to progress. Physics did this, so we should do this. Physics did large scale experiments at like centralized hubs like CERN, so we should have those as neuroscientists. But I think the nature of the brain makes it unlikely that there will be a final theory. I think it's a big mix of whatever solutions evolution could get to, and so trying to follow in the path of physics and hope that we're going to get to like cleaner and simpler descriptions that explain everything. I think that's not likely to happen. I think we need to kind of accept that there will be different theories that explain different abilities and different types of neural networks and everything, and that's fine.
But I guess, I mean, when you talk about physics, you think about high energy particle physics, and because I come from solid state physics, and there things are more messy, and it's not like one. I mean, well, when I mean, in some sense, you can say that if you're able, if you can sort of, if you put a bunch of atoms in a lattice, for example, and then you can use solve the Schrödinger equation for all these electrons in a piece of metal or a piece of whatever, and that's very difficult to do numerically, but we know that if we're able to do it, then in principle, in principle, that everything should come out of this, like superconductivity and semiconductors and metals and all these different things. So in some sense, you could call that like a theory of, but that doesn't really tell us. I mean, in practice, in order to understand things like superconductivity, you need like special theories that just like highlight that phenomena, which are much simpler. So I think maybe for me, it's more about maybe like these biophysically detailed models with large networks. That's more like this. You build that's like a large scale simulations of electrons, which tells you something because it's because you can get out numerical results which can be revealing, but it doesn't really tell you. It's difficult to wrap your head around these different phenomena. So I guess you need both, maybe.
Yeah, I mean, I think there is this question of what is the right level to be trying to understand the brain, and what are kind of we entitled to expect as an understanding, because there is this idea that came with this new wave of people using artificial neural networks as models that the kind of traditional things people were trying to do, like put individual neurons into categories and try to understand them on their own, like that was misguided, and that's never going to be how we'll understand the brain. And we need to accept that understanding is not going to look like what we thought it would, up to the point of people saying that like understanding the brain just means understanding kind of its basic architecture, what it's trained to do, and the learning rule that that trained it, and then the rest is just the emergent properties of those three things. And so that's very different than how neuroscientists have been trying to understand the brain. And
Yeah.
so yeah, but there's no reason why the way we kind of innately at first, when we could only record from one neuron at a time, like the norms that were set up and the expectations that were set up from that time in history, there's no reason why those have to have been the correct ones. And so I'm sympathetic to the idea that we need to expect things to be described on like a higher level of abstraction than people have mostly
Mm,
been working at. I don't know
mm.
exactly what that level is or how to define it or how to
I mean,
derive it
you
from neural
one
activity,
model
but
or one you talk about this free energy principle. You have this chapter which I think which is sort of fun about the grand unified theories where you sort of speculate a little bit in different directions, and so this free energy principle is one of these really high level principles that maybe can. So that's one way to. Yeah, it's one way. That's one way to phrase sort of like a theory at some level, right?
yeah, I mean, I don't have much hope that that's gonna be the answer
No.
for us, but it is
No,
something that's put
but
out there.
a principle like that. I mean, something that because that's really high level.
Yeah, see, I think even that whole kind of shape of an answer is not.
Mm, mm,
I'm not expecting to find that.
no.
Except, so
Mm,
one way you could there's one version that arguably is a general principle that you could say explains everything, and that's kind of just like the idea that artificial neural networks are universal function approximators. So just
yeah.
whatever the brain's doing, a neural network
Oh,
can do. We
true.
know that neural networks are
Yeah.
universal function approximaters,
Yeah.
and so that's the answer. That solves the brain.
Mm. Cool. I mean, of course, I mean, if you look back at the history of computational neuroscience, like, and I mean, we have always been the underdog in neuroscience. It's like both at least quantitatively, right? I don't know what fraction of neuroscientists today would sort of identify as a computational neuroscientist, maybe like a few percent. I don't really know.
Hmm.
Something like that, just by looking at the number of people going to computational conferences versus Society for Neuroscience, for example.
Yeah.
And I mean, like and for example, Ramon y Cajal, the founder of like well, it's very important neuroscientist. He didn't have much respect for theorists at all, right?
Yeah, he
And
was more of an artist, more of a observer, documenter kind of type.
yeah, but also this he sort of didn't really like sort of these people were just thinking or not doing experiments. They were some kind of lazy
Lazy. Yeah.
charlatans. Yeah, yeah.
Yeah.
And
Yeah.
there's still a little bit of that around, isn't it? I heard. I even read this. Although John Hopfield wrote a paper about his life in science, and he told he wrote there that in the 70s he was taken aside at one of these conferences by an experimentalist saying that well, this is not really good style to work on other people's data, and you have to make your own data. And I also heard like similar things in the. So it's sort of this. I just
It
yeah. So that's one
seems
thing. Yeah,
it certainly seems like it's changing though.
yeah.
You can certainly find those people who, yeah, either think that the mathematical models are useless because they don't
Mm,
replicate every single detail, or, yeah, that somehow using other people's data is either lazy or like you'll never be able to understand it. You couldn't, like, only the
mm,
person who collected the data can
yeah.
understand it well enough to use it, which is
Yeah,
scary because it's like, shouldn't you be documenting
yeah,
things
yeah,
enough
that's
that
true.
it can be
Yeah.
conveyed to other people? But certainly, I think that's changing culturally. Like, younger
Mm,
generation of neuroscientists don't see that divide as strongly. There's more people who are doing their own data collection, but also doing like serious modeling work. The grant agencies are shifting incentives to make you have to share your data,
mm,
and are willing to fund projects that use publicly available data. I mean, the Allen Institute is definitely showing this. I mean, their whole a lot of what they do is collecting data for the purposes of sharing it. So
mm.
yeah, I think things are shifting
Okay.
in a better direction.
Yeah. I just also wonder because I know the physics community, and they also felt a little bit very annoyed by sort of like philosophers of physics because they didn't really know physics, and they were sort of, oh, who are they to start philosophize on our things? It's like maybe it's like a little bit like the experimentalists thinking about the modelers. Who are they? They don't really do the
Yeah, there's a
recording,
little
so
maybe
there's
distrust or
like
something. I
or
think it
something
can
like
go I think it can go both ways though. I think that there
yeah,
are, modelers who are like, this
yeah.
experimentalist isn't understanding what my model says and what it means
No,
for their
that's true.
experiments and everything.
Mm.
So
Yeah. I mean, one goal of starting this podcast is to try to make contribute to making the computational or theoretical neuroscience community a nice and generous community. Because one thing, I mean, one thing that I sometimes compare the computation neuroscience community to sort of like trying to settle Australia with 10 people. It's just too few people it's very few people. I mean, for example, we discussed this the network that EMR has been modeling, right?
Mm.
That's not. I don't think that has been picked up by many other groups. Not because it's not exciting, but it's just so many problems, so many animals, so many ways to do modeling compared to the number of computational neuroscientists in the field. So I think it's sort of a sparse field in that sense. We are sort of often quite alone in our sort of on our own little turf. So that's sort of, so I think we have a lot of different approaches compared to the number of people we are. And one thing I've been wondering about, do you think we are a generous community that are we nice to each other?
Computational neuroscientists, I don't think we're particularly not generous.
No.
I
Which
mean,
is, I
people
mean,
build, in terms of academic things, people build like
yeah.
toolboxes that they maintain for other people to use and all of that.
Hmm.
I don't
But
think there's.
this petition against the Human Brain Project, that was a quite unique thing. That's very rare that there's like petitions against projects which has been funded, right? So that's, and that was often a lot within computational neuroscience. Also, I think that wasn't our proudest
I
hour. I
think
think,
the amount of like that funding was itself a unique thing, so I think that
yeah,
does, and also, but that
yeah.
wasn't exclusively computational, right? I mean, the
Oh,
Human
that's
Brain
true.
Project and everything includes a lot of experimental work and funds a lot of
Yeah,
experimental work as well. I
yeah,
think it's just when
yeah,
one
yeah.
entity is getting a lot of attention and resources and people
Hmm.
feel that they're not
Yeah,
representative of what the field is doing. I think that that can
yeah,
draw out, people are going
yeah,
to have things
yeah.
to say about that.
But still, it's so we should try to maybe do better and be more generous. Because if you look at these different approaches, there are really so many different approaches, and I think it's at there are not so many success stories when it comes to network behavior in particular. That we should just try out different things, and I think at least. So anyway, but I think it was really nice to have you on this inaugural podcast. So I think you had this very nice. You wrote at the end of your chapter one of your book. I think I'll just read that. You said, end of chapter one in your book. Mathematical models of the mind do not make for perfect replicas of the brain, nor should we strive them to be. Yet, in the study of the most complex object in the known universe, mathematical models are not just useful, but absolutely essential. The brain will not be understood through words alone. That was a nice sentence.
Thank
Not be
you.
understood by words alone. So
Yeah,
I
but
think
then
that should be
it's
like a credo for, yeah.
but then it is the start of 90,000 word book.
That's true. But so you need some words also, but not words alone.
Yeah.
So thanks, Phil, for coming to the podcast, Grace.
Yeah, thank you. I'm so happy to be invited as the inaugural guest.